Thank you so thank you so much. Greetings from Salt Lake City this morning. Fun to be here. Fun to be in your Zoom company. In particular, great to see Steve Goff, one of my science heroes here. So I want to talk a little bit about an evolutionary view of coronavirus diversity, and this I think is useful actually to follow on from the talk that we just heard. How do we have a framework for thinking about, as we're thinking about cross-reactivity of immune responses, how do we think about how these viruses are related to each other? And then I'll also get into maybe a point or two about forecasting, not only what we're up against with this current pandemic, but perhaps pandemics of the future as well, using this evolutionary perspective. So all of this work I'll be telling you about is being conducted by a great postdoc in my lab, Stephen Goldstein. Stephen did his PhD in Susan Weiss's lab at Penn, and she's really a pioneer of coronavirus biology. And so coming with a really good footing in the virology here, and then Stephen advancing into thinking about some of the evolutionary relationships of coronaviruses in new ways. And as you'll see, with a focus on recombination. Okay, so very familiar of everyone here, of course, with the genomic structure of coronaviruses illustrated above. And then also probably increasingly with the phylogenetic relationships here. And so in this tree, highlighting some of the alpha and beta coronaviruses, and then in particular in red, the bovine coronaviruses, the SARS-related coronaviruses, including of course SARS-2, and then the SADS-related coronaviruses, which I think are a little bit underappreciated at the moment, but are really fascinating and emerging set of coronaviruses named swine acute diarrhea syndrome, an outbreak from a couple of years ago that I'll return to sort of at the end to try to underline this at the end of the presentation. So the relationship between these viruses is depicted here based on the relatedness of the RDRP, the RNA-dependent RNA polymerase. And this makes sense. It's a highly conserved gene, and so it has some fidelity for thinking about how these things are related to each other. However, if we build trees based on other regions of the genome, the spike in particular, which of course is subject to a lot of volatile evolution, the relationships between these viruses can change pretty drastically. So here for the bovine coronaviruses, includes OC43, the common cold, and others in that category, you can see just by the color coding that it's been shuffled a bit. That's because the sequences are quite different, even relative to the RDRP. Same for the SARS-related coronaviruses. You can see that little blue cluster is now spread out, RMYN02. It now looks like an outgroup all of a sudden, where it didn't before. And I'll zoom in on that in a moment. And then we'll kind of think about this emerging cluster as well, which is almost like a rainbow of diversification when we look at recombination in particular. Okay, so we were really interested in that incongruence of phylogenetics, depending on the part of the virus genome that you use to try to understand the relationships between them. Kind of ran into a wall, which is it's really hard to map that relationship between recombination and phylogenetics. And so we set out to develop a tool to help to tackle this complexity. So the tool is called IDPlot. This is what I'm describing is on the bioRxiv. This is also open source software available at GitHub if you search for IDPlot. So basically the workflow here is to take the coronavirus sequences or any sequences you'd like to compare to do analysis of recombination. In red are the tools that this puts together into a workflow. All these tools we didn't develop. These are existing tools. So MAFT provides you an alignment between the sequences, which is then that alignment is used by this tool GUARD. And this is part of the phylogenetics package, which actually predicts or proposes recombination breakpoints based on your alignment data set. Once you have these proposed recombinant regions, then this program FastTree, based on that, will take each of these separate regions and then build a different tree, again, based on those relationships. So this is what this workflow looks like. And I'll go into a little more detail in slides ahead, but the GUARD refinements and you can actually sort of dynamically pick how many recombination breakpoints. So if you look down the plot at the guard, kind of, if you tilt your head at 90 degrees, those are the recombination breakpoints predicted from these sequences. And then a little bit of the details that will change depending on how you allow the statistical model to try to become more and more complicated or to fit the data more closely. And so this pipeline, I should say, is automated, but then really the user, depends on the user to sort of customize or to compare the sequences or the questions to really come to some understanding. So it's kind of a little bit of science and art together. So then below that guard, you have the ANI. This is the average nucleotide identity. And it's relative to your parent sequence that you choose, and then comparing it to other sequences. And you can see, for example, that orange squiggly line shows equine coronavirus, the differences in nucleotide identity compared to bovine coronavirus. And so those dips are pretty diagnostic of recombination regions, although it can get more complicated. So you have one, on that left panel, you've got one recombination region called, but there's clearly 3 other knock-on regions, those additional dips in the equine coronavirus. And so you can fit that by going across the refinements. And then the trees are actually drawn based just on these sort of compartmentalized genomic regions that you're proposing as recombination regions. Okay, you'll see this again a little bit here. So this is now not the bovine coronaviruses, but the SARS-related coronaviruses, SARS-2 at the top. And so these vertical ticks, those are just the RNA nucleotides. And then the ticks below are ones that are different from, in this case, SARS-CoV-2, the reference sequence. And so here for SARS-2, it's a little more quiet than the bovine coronaviruses, but you can see recombination regions that Stephen is proposing that correspond to the spike region. And so this isn't a new finding, but does show that IDPlot pulls out what other people have seen or proposed. But there is an important point here to pause on for a second, and that is that having the evolutionary framework allows you to reconstruct when and where these recombination events happen. And so there was some misidentification proposing that SARS-CoV-2 actually underwent recombination, but even relative to rat G13, the most closely known related virus a few months ago, you can see that that's not the case, that it's some of these other pangolin and bat coronaviruses that have undergone recombination. And that's shown by those dips that then you can use for phylogenetic analysis. So here again is this sort of phylogenetic shifts, depending on the region you're using, will really dictate the relationship you infer about the viruses. So the RDRP on the left, which I've already shown you. And now if you do this by recombination region, you can see that the bat coronavirus, RMYNO2, which is the most identical for RDRP, it clusters most closely with SARS-CoV-2, now jumps out as an outgroup. And then a similar thing, if you look at ORF8, the somewhat mysterious and I think underappreciated gene, probably involved in immune evasion, and is often deleted in some of the viruses. It's been proposed that that could actually quiet down the virulence of the virus in some cases. And so this is, I think, another important point to consider, is that it's really not just the spike protein and recombination or diversity there that's dictating a lot of the diversification or evolution of coronaviruses. There are a number of other regions throughout the genome, actually, where this accelerated or I should say high rate of recombination is really influencing coronavirus biology. Okay, so I wanted to spend a moment on this sort of, I think, underappreciated in some ways set of coronaviruses, this lineage SADS. And it was in like 2019 or so, an outbreak on 4 farms and leading to the deaths of something like 25,000 pigs. And then a little bit more like SARS-1 than SARS-2, sort of just disappeared or fell off the map, hasn't gone under, or sort of lurking perhaps in the shadows, with some potential perhaps to spill over to humans at some future time. And so if we look at the SADS coronaviruses, now 8 comparisons here, It's a little bit like recombination on steroids. So not only in the spike region do we see a really complicated recent history of recombination, basically an event or more for each of the sampled viruses here. Also, recombination regions quite clear near the 5' end and the ORF1ab, some of the more structural or conserved parts of the genome. That is sort of a general theme, is that both 5' and 3' ends seem to have more recombination events. This could be for several reasons or overlapping reasons. I think a working hypothesis would be that recombination is actually pretty prolific across the genome, but then selection acts to retain diversifying events closer to the sort of distal edges of the genome. Okay, but so now if we start to look at all of the recombination regions and how that changes the phylogenetic relationship between the viruses, it's all over the map here. And so this is of course not meant to look at each detail here, but to illustrate that these viruses are really sort of a chimeric collection of different sequences that are sort of cut and paste together. And so we're thinking now, we don't have any answers, but are thinking about how do we even represent these viruses. Any one from an evolutionary standpoint, any one tree will only be true for a small segment of the genome. And so the RDRP, of course, makes some sense, but as we better understand the biology or the important points about how these viruses are changing, those relationships will be scrambled. And that's something that I think will inform, the better we can understand that, the more it will inform how we understand things like cross-reactivity of immune responses, other sort of practical considerations like that. So another way to sort of simplify this and look at it, which illustrates, I think, another point, so I'll get to in a minute, is to just actually very simply look at the spike regions, S1 and S2, divide them, the spike in half, and then do very simple blast searches. What's the highest identity as we just take out these little fragments, these recombination regions that we're proposing through IDPlot, and then asking who are the closest relatives just from a in this case, an amino acid identity. And in red illustrated there are some surprisingly low numbers for the highest match of the sequence, that fragment, from some of the SADSr coronaviruses. So if you're around 70% identity in a spike, what's happening here is these viruses are basically as we're sampling them, are returning to us the fragments of other viruses they recombined with recently that we've never seen before. And so I think what this really illustrates is how sparsely sampled a lot of the coronaviruses are, not only in these sort of less well-studied clades or lineages, but of course all the way through SARS-2 as we grapple with the origins of this virus. And so it's almost like these viruses are sort of genetic probes bringing back hints about some of the diversity out there. And I think maybe sending a warning signal that there's a lot of coronavirus diversity that we are just barely glimpsing at and should probably endeavor to know a little more about. So just my kind of take-home messages here is that we and others are, I think, seeing really clear signatures of extensive recombination, and this probably reflects somewhat clinically rare or sort of among animals rare co-infection events, given that these fragments in some cases have so little relationship to other viruses or limited relationships to other identity to other viruses that we've seen. This implies that co-infection is happening even among very diverse coronaviruses within lineages that we're still sort of glimpsing at. And then, in sort of a related point that I think this also highlights the limited sampling that we have, or the limited understanding of all of the diversity out there in wildlife and other sources. Okay, so I'll stop there. The creation of the tool IDPlot was a really fun, or is a fun ongoing collaboration with Aaron Quinlan's lab here at Human Genetics at Utah, and here's sort of a hybrid view of the LD lab stepping through the pandemic. So thanks everyone. Thanks for having me. Happy to field any questions if there are some.
Okay, thank you very much, Nels. This obviously was much faster than we expected, but great for a presentation.
Oh, I actually lost track of time. Oh, I'm 3 minutes early.
And
we can all, sorry, go ahead.
No, I'm just checking whether there are any questions from the audience. I see that Steve Goff has a question.
Okay,
Go
hey
ahead.
Nels,
Steve.
good to see you. So I have a general question sort of about the software, I guess, or the program and or how we interpret things. So when do you make a call that a newly appearing diverse sequence was due to a recombination versus just some high pressure response? So, you know, when we see the UK spike appear, we think of it as rapid evolution, 10, 20, whatever new mutations appearing, you know, in very short time. And you would say, well, maybe it was recombinant, maybe it was just rapid evolution. How do you make that? Where's that cutoff made?
Yeah, it's a great question. And so, and it's kind of, you know, it's a continuum. So you can make high confidence calls in some cases, and then it's sort of, you know, both diversification mechanisms are possible as well. And so certainly the very recent evolution you're describing, some of the variants of concern that are emerging for SARS-2, these actually can fit pretty neatly into a collection of point mutations accumulating very rapidly with selection acting on them. So, but if we look here at this, at some of the bovine coronaviruses, for example, so just at the 5' end, so the orange virus that has that kind of extended region with 3 dips in it, this kind of illustrates a clear example of a recombination event, right? So you have all of these tick marks that are the reason that you have that dip in the identity. And then there's that break, and it's basically identical to the equine or coronavirus is identical to bovine coronavirus. And you could say, oh, well, wait a minute, maybe it is still just that it's diversifying like crazy in that region, but you're so constrained in the other region that this could still not be recombination. But then when you look at some of the close cousins here, OC43, for example, right, you see, well, actually, no, you can, and PHEV, you can actually tolerate all kinds of mutations there. That just hasn't happened in equine coronavirus. And so that's a pretty clear, you know, differentiation between those two mechanisms. The other way to diagnose it a little more at the edges is the idea of hitchhiker mutation. So if you have those, like say 20 changes in one variant of concern relative to another, and then you propose, oh, there was a recombination event between these two, that you would base that on the fact that you now bring together, you know, these unique mutations in both of them. And that would be one evolutionary event versus the 20 evolutionary events it would take to see that combination of hitchhiking mutations or sort of companion mutations with the ones that might be selected on. And so that's just, it's a parsimony or an argument of parsimony that it's simpler to do that in one evolutionary step than in, you know, 20 or 30 evolutionary steps. So right. That's kind of how you work through it.
The smoking gun for that in a way is to find the other parent. I mean, if you see that's the right. Other parent that has all of the stuff that appeared, that would nail it. Yeah.
Absolutely right. And again, I think that illustrates the issue of limited sampling, right? So our inferences are only as good as the samples that we see. And so, and the more that are out there, it really, the inferences become much more powerful.
Yeah, thanks.
Nels, very nice talk. And my question is a slight follow-up to Steve's, because I want to ask a little bit about the mechanism. Do you feel that the recombination events are just due to template switching, which is the way people think about recombination in these viruses? And how much does the error-prone transcription really play a role in giving some of these multiple events?
Yeah, it's a great, thank you, really good question. So maybe the last one first. And so one of from an evolutionary standpoint, evolutionary virology, you get a little bit sort of confused given the proofreading. So large RNA genomes, these coronaviruses are kind of super tankers of the RNA gene, and need this proofreading, we assume, to not just completely crash as a virus population. And so that's where I think the recombination is coming in to sort of pick up the slack of the diversification you need to compete with other viruses, to compete against a diversified immune system that you'll see in varied hosts. So template switching probably works. I think one thing that's underappreciated, this is work of Mark Dennison, I think, which is really worth keeping an eye on, is in addition to template switching, all kinds of virus forms that aren't complete in the population. So recombination happening where you're actually truncating the virus. In many cases, these are not going to be productive particles. But in the context of a big infection or a rare co-infection, another source of diversity to sample from. And so it's worth a little bit of caution in interpreting that data, just with the potential for long read sequencing, deep sequencing to sort of create artifacts. But I think we're far enough along that we can say that in addition to sort of traditional template switching, that you might envision that there's more going on here, that these things are sort of spinning out all kinds of fragments, truncated fragments that could be sort of the fuel for doing more recombination more quickly.
I could make a comment to Rodney, just that,
Yeah.
I mean, 2 points that we know the polymerase is very good at hopping. It actually has in minus strand synthesis, you know, these directed hops. So it has engineered into it hop capability normally. And I'm sure if all we have to imagine is that it hops, you know, inappropriately, and that would do it. All of those hops are intramolecular in the utility for the virus, but of course, if there's 2 viruses in there, you could easily imagine it going intermolecularly, and that would do it. The other thing is, I'm amazed there haven't been a lot of experiments where people co-infect cells with 2 distinguishable coronaviruses and directly looked at recombination. And I'm sure that's in the cards to be done.
Probably a good idea that people don't do that so much.
Actually, it's true, getting permission in the potential gain of function environment that we're in is an issue. Yeah.
Yeah. No, thanks, Steve. Really good points. And but and also, I think it is in that gain of function sort of perspective. I don't want to put too much gas on the fire here. So just because you're recombining doesn't necessarily mean that you're recombining with sort of, you know, super bugs or something like this. Most of, just like evolution as usual, most of this recombination will lead to fitness defects in the products, or will be quite neutral. And so I think there is, you know, no reason to sort of, we're in the sort of current situation that we are in. But I agree, it's a really cool experimental, it's a great experimental idea, and, you know, taking those precautions to think about and to set up model systems actually, where we know that there probably is little, if any, you know, potential for a gain of function, and then, but to understand the rules of how these things actually do these mechanics, which is
Nels, if you set up a recombination assay, I'm super interested. So
cool, thank you. Yeah, we're so stay tuned, Stephen. This is on his menu, but for Stephen Goldstein's menu, but for reasons of for moving slowly.
all right, yeah, it's tough. We understand. All right, thanks a lot.
Thank you.
All right.